The overall goal of this procedure is to demonstrate simple and rapid methods for performing FISH on endosymbiotic bacteria and viruses in tomatoes and in the whitefly Bemisia tabaci. This is accomplished by first sectioning the tissue by hand, then fixing, or dissecting the internal organs of insects. After the fixative works on the prepared tissue, the samples are washed and hybridized overnight with the respective probes. The final step is to mount the samples and view them using confocal microscopy. Ultimately, virus and endosymbiotic bacteria can be localized in plant and insect tissue via a simple hybridization and use of highly specific, short, fluorescent probes.
We first had the idea for this method after we localized endosymbiotic bacteria in Bemisia tabaci. The main advantage of this method over existing methods, like using antibodies or riboprobes, that the hybridization is simple and it does not involve complex steps like probe design, washing, and handling steps. Demonstrating this procedure will be Adi Kliot, a graduate student in my lab.
There are three important preparations. First, rear B and Q biotype whiteflies on cotton seedlings of the Acala cultivar in insect-proof cages. Second, check the endosymbiont infections by PCR with endosymbiont-specific primers. Third, obtain beefsteak cultivar tomato seedlings and rear them under the same conditions as whiteflies until the plants reach the 4 true leaf stage.